we're a bit of a mixed audience, so I'm going to give you a tasting menu. And I think this talk's a little different than what you've been hearing about. The focus is going to be to have you leave here with a feeling of what we're actually doing in the clinic as you guys, work in the pipeline coming in, the technologies coming in. This is basically where we're at. And I'm going to take two trials that we have, trials/therapies that we have ongoing at sick kids that basically started up in the last 2-3 years. So this, and we're sort of at the vanguard in Canada in giving these cell therapies to kids. When I use kids, I mean adults too. So that's we're non-discriminatory. So I do want to disclose that I'm working on clinical trials with CRISPR and Novartis, none of which will impact this talk. I have no financial issues. So the goals are just very simple, to give you a taste of what we're doing and how we grew from basic understandings of physiology to take things forward into clinical practice, and to give you a flavor of just what the heck is involved in finally getting patients treated from time that we have a good product to getting it so that we actually make kids better. Our cell therapies are based upon really fundamental hematopoiesis. Our blood making is driven by a small number of primitive hematopoietic stem cells that live in our bones. They give rise to both the immune system and the hematopoietic system, and we take advantage of the fact that if we can make a change in the hematopoietic stem cell, the really primitive hematopoietic stem cell, that we can affect long-term production of anything in the red cell, white cell, platelet lineage. And then secondarily, with the CAR-Ts, we're picking out these guys, the mature lymphocytes, and we're putting a little Velcro on them and getting them so that we can get them to do their job, and their day job is to get rid of things that don't belong in our body. So they're the professional killers in our body. So those are the two themes that I'm going to talk about. We have a gene therapy trial that is open, looking at correcting what used to be a lifelong disorder of red cell production called thalassemia. Specifically, we're looking at beta thalassemia, which refers to a specific subset of patients with thalassemia. And basically, what makes our blood red is the heme ring and the hemoglobin protein. And it's actually a common disorder where we make too little of the hemoglobin, and we have an imbalance in the subsets that build hemoglobin, and the net result is that you break down your red blood cells. The net result is you don't make enough red blood cells, and that would make you very pale, not survive out of childhood if we didn't give you lifelong transfusions. And if we give you lifelong transfusions, we're pouring iron into you. Too much iron is a poison to the body, causing a whole host of its own problems. As part of getting into this gene therapy trial, I work very closely with patient focus groups with adults who have been living with thalassemia, just very humbled with the amount of morbidity with this disorder. Because if you have you have to come to clinic every 3 weeks for transfusions, 3 to 4 units. That means you're not traveling very far from your healthcare center. That means you've got a lot of iron you have to get off of you, so a lot of monitoring, and the drugs to take iron off are tough to take. But what the families complained about and the patients complained about was the bone pain. and the chronic back pain, knee pain, and basically our blood is made inside our bones. You expand that bone cavity, you run your physics of us being bipeds, and you get, spine problems, joint problems, and just a lot of pain and suffering with this disorder. Similarly, sickle cell anemia, also a problem of the red blood cells of the same part of the hemoglobin, the beta chain. One single amino acid change, one that is caused by a TA switching to an AT here, results in sickle hemoglobin. Gives you a survival advantage for malaria if you have sickle trait, but if you have sickle cell disease, basically 2 doses of this, your red cell, and it's straight physics, van der Waals forces, causes your hemoglobins to line up and make, instead of having the donut for your red blood cells, you have spikes on your red cells, they get stuck, you get strokes, you get bone pain, expansion of the bone, joints, problems, big lifelong suffering. So what these first trials are doing are taking advantage of our fundamental knowledge of how hemoglobin is made, and that there's on 2 separate chromosomes, there's an alpha globin gene and a beta globin gene. And then for years, basic fundamental biology studies looking at something that seemed like esoteric research that, why are these guys studying this? There is a change that happens between being in utero to being out after birth, between the kind of beta globin gene that you make, what's actually called gamma globin, or fetal hemoglobin, that happens at time of birth, probably related to us coming out and breathing a lot of oxygen, associated with the switch. But there's a natural physiologic switch that happens at time of birth. The defect for thalassemia and for sickle cell are in the beta globin. The gamma globin gene is fine, okay? So if we could just keep these guys in baby state, maybe that would be sufficient. And so this picture just shows you what happens normally in development, that at birth there's a big switch between making gamma globin and to beta globin. Kids start presenting with sickle cell symptoms and thalassemia symptoms at about 3 months of age, when you've got most of that switch already happening. Here's where we went to the friggin' epidemiologists, the guys who are out in Africa collecting data, the people who were looking at, curious families, case reports and series. And we found that there are some families who are walking around who, by just the luck of the draw, have thalassemia, have sickle cell disease, but don't make that switch to fetal hemoglobin, from fetal hemoglobin production. Lo and behold, there's even some families in Saudi Arabia who don't make the switch at all. Normal lifespan, much, much lower to no symptoms from the thalassemia and the sickle cell disease. We know genetically they've got the disorder, but they're asymptomatic. So we have the clinical picture of, gee, this should work. If we can just make these patients fetal hemoglobin producers, we should be able to treat beta thalassemia, we should be able to treat sickle cell. So that's what this is showing. The goal of our treatment is to move patients from, to make them look like the people who normally have persistence of their fetal hemoglobin production. How can we do this? Well, again, the basic biologists in the laboratory spent a lot of time looking at transcription factors. What makes that switch between fetal hemoglobin to beta hemoglobin production? Research where you would say, where is this research going? Why should this research be funded? Kind of stuff. And here's why. There is a complex, transcription factor complex, that sits and prevents the production of gamma globin genes, okay? Key component of this is a transcription factor called BCL11. We know that families who have defects in BCL11 have high fetal hemoglobin levels. They're in that cohort. We now bring in the new tech tools that we have, and so using CRISPR-Cas9, we take a guide sequence, an RNA that matches the part of the BCL11 gene, and we use that as our guide sequence, and with the caspase, which is an enzyme, everyone has to go like this when you go CRISPR-Cas9, you go like that. And what we do is it's actually very elegant. We take the young blood-making cells, the youngest of the young blood-making cells, we select them. We give them a little electroshock therapy with electroporation. This complex of CRISPR-Cas9 with the specific guide sequence swims across. It's an enzyme reaction. It happens in a short time period, a couple of hours. It's done. No virus transfect needed. And what the first generation of treatments with CRISPR-Cas9 are going to be and are ones where we disrupt the gene. We know that down the road we're going to be able to build new, to do more elegant things, but right now this is kind of a first-in-human approach. So we're coming in, we're going to remove the BCL11 activity and prevent the switch to beta hemoglobin production. Okay, nice in the test tube. Okay, beautiful in the test tube, but is this going to work? And what are the safety issues? And so this is just to remind me to say, yeah, that's nice if I correct the cells that want to make red blood cells pretty soon. These guys in here, they live a few weeks. Okay, and then they die off. They have the terminal differentiation signals. In order for this to work, we have to prove that the most primitive of primitive cells are transfected, and that's going to be the acid test of this first trial. As best as can be done, using colony assays, using immune-deficient mouse transplant models, it looks like we can correct the most primitive of primitive cells. We have to show that we don't, by accident, take something else out at the same time. So extensive work's been done looking at where this 15-mer guide sequence also binds into the human genome, because we don't want to cut out something that is needed for something else in the blood-making system. And as best as can be modeled, there's nothing that is going to get taken out. But as part of this first-in-human trial, we are looking very hard for anything unexpected, and patients will be followed for at least 15 years afterwards looking for the I don't knows. So what does this clinical trial look like? We have the first few patients enrolled. The first 2 patients worldwide have been treated, and I think we've got patient 3 and 4 coming on board. And so what we have is we have our patient who has thalassemia. We're collecting their young blood-making cells through a process called apheresis. We coax the young blood-making cells into the circulation. This is our day job. We do this in bone marrow transplant all the time for other reasons. We know how to do this. There's a few things we do know how to do, and there's a lot we don't. But that one's safe. And then in the laboratory, we pull out the young blood-making cells that have hematopoietic potential. Those are the CD34 positive cells. Those cells, and this is the picture for electroporation, they get a little zapper, and the CRISPR-Cas9 goes in. They are made so that they won't make that switch. We freeze the cells. Cells are shipped back to us. Now we got to get the patient ready. If I just poured those cells in, the patient's own blood-making cells are going to say, it's like having a garden with weeds. If you just pour seeds onto a garden with weeds, nothing's going to grow. So we have to use the defoliant, and so that for us is a drug called busulfan. It's a chemotherapy drug. We're not proud of it. We need to do this better in the long term. But once we use the defoliant, we then infuse the young blood-making cells. The gift we have is that if we put young hematopoietic stem cells into the vein, they know how to get back into the bone. Okay, so there's no surgery involved. We wait for the blood counts to recover, and then hold our breath. So this is doesn't go anywhere. But the first patient is now who was treated is now transfusion independent for close to 9 months, and okay, that's fine, but that's not where we want to be. But so it's at least we have a signal that all this is in the right direction. There is another strategy that is that you guys may have heard of. That is we're working with a company, Vertex CRISPR Technologies, an alternative approach that was recently published. Uses technology by Bluebird Bio that puts in and uses retroviral gene therapy strategy, putting in a beta globin gene that's corrected, and looking at correction of blood making in the same patient population. So I'm a pediatrician, and the red bars are the pediatric patients, and so all I care about are kids. And this time with the corrected beta hemoglobin gene can be measured in the blood, so just simply taking blood hemoglobin measurements, and they can see that they're making sort of 10 grams per deciliter. It's a hemoglobin of 100 in Canadian terms is fine, okay? So these patients are making off of that transvected hemoglobin. So it's not perfect. Patients with more severe forms of thalassemia have had less transfusions and less iron issues, but and so it's definitely an area that needs more work. But first proof of principles are incredibly exciting. What I just wanted to kind of just give you a flavor of from our end, there's a lot that goes into optimizing the vector, the correction, what's done in the laboratory, but there's a whole ton of other variables on the clinical side. How many of these cells do we need? How do we best deliver them in? Should we put them into the marrow cavity or just into the bloodstream? Is there a difference with age and disease? Do we need to hit a sweet spot? I'm already we know that our conditioning with busulfan is not great. I'm going to give you a little just switch to CAR Ts. So the other new kid on the block that we're incredibly excited about are CAR Ts. And so CAR Ts are, and I alluded to, Velcro T cells, so that T cells are professional killers. We're getting them to the spot where they need to go and getting them cuddled up against the guys they need to do their job on and getting them excited. So T cells normally have on their surface a T cell receptor that will recognize something as foreign, such as a tumor cell, with a tumor peptide and get rid of the tumor cell. But when we're treating kids with leukemia, we know our own immune system's flunked. Okay, it can't see it as the leukemia is foreign, it doesn't have enough jam to get rid of it. So what we've observed is that if we take the other part of the body, the B cells, the human B cells, give us lifelong immunity. You get your tetanus vaccine as a kid, you've got immunity for tetanus for decades. You probably need a booster and yada, yada, yada. So what we've done with this technology is take the recognition component of an antibody, it's brilliant, and so you need to have something on the surface of that cancer cell that is on the surface of the cancer cell, not on the surface of other cells that you need to live with, that has enough on the surface. So there's just a lot of nuances in what I'm saying. But if we take that antibody recognition site, put it onto a T cell, we now have a professional killer cell that's got the ability, if it bumps into a cancer cell, to hook up, get activated, and kill the cell. Now, the first 20 years of work on this, a lot of big studies, famous people, were duds, okay? Nothing, no action, minimal clinical activity. And that's called the first generation CAR T cells. And so there they had their recognition site. So they knew they could show they came up against the leukemia cells, but they weren't able to kill the cells. So what the guys at UPenn and CHOP did is they added into the construct a co-stimulatory factor, something that says, I engage, I can activate the T cells, but when I activate, I want to get other juices going in the T cells to make them better and to get them to go into cell division. So those are called co-stimulatory factors. And then the next generation, third generation, we're adding in more co-stimulatory factors. We're adding in extra targets, so dual target CAR T cells. And so these are just starting to get into phase 1 trials. But what we have right now for us clinically is called the second generation CAR T. And so it's really just as simple as this. Or when I talk to the kids, you got the X-wing, Y-wing fighters. You really want to go with your Millennium Falcon with a good amount of Velcro on it, have it hook up and bounce up against the Death Star, drill a hole in it, put the payload in, and then get out of there fast, okay? So in Star Wars terminology, those are CAR Ts. And the sad thing, I was talking to a young teenager, a preteen, and I'm starting to go through my Millennium Falcon, and he's looking at me. He said he's never seen Star Wars. Like that is like it's awful. Like I just don't know. Okay, so here just quickly, I just want to give you a flavor of what happens to us on the clinical side, because for us to pull this off clinically, it sounds quite simple. You take the T cells. You correct them, you do a little gene magic with the lentivirus in the lab, and anybody who's a lab rat says it's a 2-hour kind of thing, no big deal. Freeze the cells and then go, it's a big deal. And it involves us, because for the first time, if we're looking at a pharmaceutical process, we're providing the raw material for a drug, the CAR T cell, from that patient. So that means we now have to think like pharmacy companies and be compliant with a whole bunch of stuff. We collect the T cells, but remember, these patients have leukemia, okay? They've had a lot of chemotherapy. It is very hard for us to find the sweet spot to get cells collected. And remember, we want those cells to be active and functional. And so that is also is tricky, because these guys have leukemia. They've had years of chemotherapy. We're having a lot of trouble getting cells for manufacturing. We need manufacturing slots, okay? We need a company that's ready. We're sending cells on Tuesday. We need them to be able to manufacture for us. In between, we need to keep the kids alive. And that is non-trivial for us. We then give them just before we hear from the company, we got the cells, they're working, they look good. We then give them chemotherapy. Again, the same idea, making space. We get rid of the weeds. They're old lymphocytes. We want these new lymphocytes that are really targeted to have extra space in the body, ability to grow up. And then we give them. Now, and these new second generation CAR Ts have the signal to grow. So you give 1 million cells into the body, they bang up against a leukemia cell, they kill that cell, they divide. So suddenly we have a ton of cells kicking up a lot of background byproducts called cytokines, and the kids can get very sick. And so about a quarter of them will end up ICU sick with it looks like call it cytokine storm, super flu, high fevers, low blood pressure, and you can get some brain problems. And then after that, CD19, which is our target, clustered on the leukemia cells, is also expressed on B cells. That means we take out their B cells, and so they need immune globulin as long as those cells are around. So we have, for this audience, I'm going to just go quickly. I kind of talked through this slide. There are a lot of things on a clinical end that we've had to train the ICU teams, the emergency room teams, our referral teams, our own care teams on how to handle these patients. We're getting better at it, but it's not for the faint of heart. it works about half the time, maybe a little better than that. Okay, we get remissions frequently, but there is a loss of the CAR T function, exhaustion of the T cells, and burnout. And then cancer cells are just friggin' smart. They know how to modulate, or you select for leukemia cells that have modulated the CD19, change to just a little bit where that antibody binds, and they'll escape. Some of them, many of our relapses are CD19 negative. They just get rid of it on their own. So we're still very much in early days. And then we're seeing late, basically lumps of leukemia that come back years later. And so we're still some hidden spots. We're still got a lot to learn about this new therapy. Treating kids who otherwise were untreatable, incredibly rewarding, still very early days. So in conclusion, I just want to highlight the importance of basic science in that unsupervised curiosity, to lay on a theme that's seems to be running through the afternoon, to just explore why, and then bring it around full circle and apply it clinically. And then the most humbling thing for me was the importance of engaging the patient and family community in this process, because you're asking a lot. And when we have a patient who, we're going to, sort of lovely young woman with thalassemia, we're offering potential of curing her disease. But by the way, you're going to lose your hair. By the way, it's going to take a month in hospital. By the way, we're going to muck with you. And so you have to really have a good it's a partnership. So just thank you, and I'll answer any questions.